Greetings, I'm Salvador Cordova. Welcome to the Evidence and Reasons for the Christian Faith video channel. I am privileged to be joined by Anthony. We're going to have a tutoring session on whatever he thinks I can help him with, and I'm privileged to be of service to him. I've been encouraged to start a speaking and teaching ministry, and I'm now semi-retired, so I'm freed up a little bit to do this. And so this is a way of advertising the ministry and also showcasing how I might be able to be of assistance to individuals and congregations and communities around the world. I know of one person that kind of started the same way that I'm starting out. And after years of doing this, he now gets to fly 77 times a year because he's in so much demand. His name is Jay Seegert. He's been an inspiration to me. For those, I think a lot of you know who I am. I'm a molecular biophysics researcher. I've had a lot of fun in life, and on some level, I've never grown up, which is kind of bad because I've just been having too much fun. And man, sometimes it sucks to have to deal with responsibility, but I'm, better late than never. I feel that we learn from each other. There is a verse in Romans, I believe, chapter 15. We are able to teach one another. So even older men and younger men can teach one another. Women can teach each other, and we can all learn from each other. I complimented Anthony on how physically fit he looks, and he was giving me advice, offering advice on, how to work out. And at this stage in my life, after I was diagnosed a couple years ago with pre-diabetes, the doctor said, look, it's not bad. An improvement in nutrition and exercise, you'll be fine. My mother suffered diabetes, and I took care of her during that ordeal until she went home to be with the Lord. I don't want to go down that path. And so I'm very grateful for people who lead by example and taking care of themselves. And I hope we can all start living better. And one way that I really do like to teach intelligent design and creationism is to talk about nutrition, because so many of the chemicals we study, when you look at all the the ingredients and the foods you consume, this is a great opportunity. It's also a great opportunity to learn how to combat cancer and diabetes and other sorts of ailments. So I just wanted to lay that on the table. I have found that young men who become Christians are not fed adequately in the churches. They have questions that are never answered. They're just dodged. They keep coming to church hoping they'll find an answer, and they don't get answers to simple questions like the problem of evil. What's the evidence of God? Why he's so hidden? To that end, the name of this channel is Evidence and Reasons, but I'm writing a couple books. One, Darwin's Dastardly Deception, and the other is Finding the Hidden God. And the theme of finding the hidden God is something that's close to my heart. It's something I struggled with for 45 years. Because God is not as evident to our senses as the air we breathe. And so why is that? And what are the cures for that? How do we deal with these facts? So with that, Anthony, I'm here at your service. I kind of gave my spiel of what I'm about, and hopefully this isn't the last time we meet. So we'll go as long as we can go before we're exhausted. And if we decide to reconvene, I would certainly look forward to that.
Oh yeah, certainly, Sal. I would definitely love to do this again. I guess to get us started, in terms of your specialty of molecular biology, one thing that I've kind of been itching to get a bit more information on is everything you cracked about the whole Nylonase bacteria, everything you did with that, because I've tried to read through the technical publications, and I do the best I can. But we both know how hard that can be to follow in a lot of ways. So, maybe just some tutoring on that. That would be some information I'm really grateful to get.
Okay, this is going to be good. The other thing is, my understanding is, you probably not had college level biochemistry, chemistry. That's fine. How about biology? Any any biology in high school or
Oh, well, yes, in high school. Yes, yes.
okay. This is. I'll tell you the good thing about learning in biology. God does not make biology so complicated that um, a high schooler cannot learn about creation, and a high schooler cannot learn that evolutionary theory is wrong. Um, there is a motivation by evolutionary biologists to not make their arguments clear. They give the illusion that it's so sophisticated you cannot understand it. But when we actually study it, realize you can't understand it because it doesn't make sense. But they couch it in this language. So my goal then, I'm sorry that we're going to have to. I'm going to have to ask you some questions about your level of understanding, and I'll try to tune my response to you on your level of understanding. So, are you familiar with? Uh, can you tell me what you think a protein is? And as you're talking, if you see me typing and you hear me clattering the keyboard, it's because I'm desperately trying to get images that I can maybe illustrate the point. So why don't you tell me while I'm trying to search this, tell me what you think a protein is.
So, to my understanding, protein are more or less the physical blocks that make up our microbiological structure. They're sort of the biggest portion of that. You get things like, uh, one of your favorites, which was the topoisomerase. There we go. I lost the word.
Isomerase.
I really liked your videos on that. I really liked how you explained that they're necessary to untangle. They have to cut and then reattach the DNA because otherwise they get supercoiling, which would be a big problem on the transcription level because it just wouldn't allow it to happen. They're made up of the amino acids, which are coded via the DNA. GCTA gets put in the correct sequence, and then said correct sequence folds up properly to be the protein that needs to go be put wherever to build whatever enzyme or skin cell or what have you.
That's really, really good. Okay, so I hope you'll forgive me, Anthony, if I don't always directly answer your question. I'll try my best. There are people that are tuning in, and so sometimes, even if I know you know something, I might just review it. So, thankfully, because we eat foods, we hear the word proteins and carbohydrates and lipids or fats. Often, most of your nutrition stuff hardly ever talks about the other major chemical, which is nucleotides. But since we hear the word protein a lot, it's not so unfamiliar. So meats are rich in proteins. And we have protein shakes. Bodybuilders like stuff with a certain amino acid, and we can go there. But so, oh yeah, whey protein. So just to review, we have the mRNA. Okay, so a protein it looks like this beads on a string. It doesn't really look like that chemically, if you were able to somehow see the atoms, but it's a good way to conceptualize it. And so the protein is made of amino acids, and there are 20 canonical. There are actually 22 total, or even more. But the most essential, what they call the canonical, there are 20 of them, and they can be represented by alphabetic letters. I wish they'd actually used the alphabetic letters here just to emphasize the point. So spelling, you can spell a protein in that it's basically this long chain. Now, amazingly, oh, by the way, Anthony, if I say something that needs clarification, feel free to interrupt me. Is that okay so far, what I described?
I'm tracking on everything you described so far.
Okay, that's great. All right, so I need to show something about the shapes of proteins. And this will relate to the Nylonase. And I'm sorry that I'm having to not, people aren't like, well, Sal didn't directly answer the question. It's like, I need to make sure everyone understands where we are, because I could answer it in my technical terms, and then, I risk people not knowing. So there is something, I'm looking up a diagram to show how differently shapes. So I'm Googling different protein shapes. And this is going to be critical to understanding. Yeah. Here's a picture. It's mostly conceptual. Let's see if I can get this. All right. Let me bring this up. So we have the spelling of the amino acids. Now what happens is all these proteins have different shapes. And the spelling changes the shape. This is amazing. If you spell the amino acids, put them in a different sequence, you get a different shape. You get a different function. And there are a variety of them. It is unbelievable. Now, some people, Anthony, have you heard the term, hang on, let me get this back on. Have you heard the term protein folding problem?
Yes. As far as I'm aware on that, it's the fact that there's a lot of proteins, while you might have the correct spelling in terms of the amino acids, they require a separate enzyme to come in and manually fold them to the correct shape so they can actually perform their function. And that sort of creates a bit of a chicken and egg issue. Am I off base with that? I mean, I know that's a thing, but is that what you're referring to?
It's part of it. It's part of the issue. Definitely, sometimes you need these chaperones or whatever processes to fold it in the right shape. But sometimes some proteins will spontaneously adopt the right fold or shape without any help. And so there are two things. The spelling here, but they use the word peptide chains. Okay, so I'm sorry for the terms. Some of the terms just kind of have historical reason for why they're named, and sometimes, so you'll hear sometimes we'll talk about proteins as a polypeptide. The same thing. I'll try, if I start using terminology that sounds that confusing, just point that out. And we'll say, okay, polypeptide, protein. It's not exactly the same thing, but close enough. So the spelling of the the sequence of the amino acids, which I call spelling of the protein. Spelling of the protein. Why don't I just show what that looks like? So we mentioned, and I'm sorry to have to do this live, but we're kind of doing this on the fly, this tutoring session on the fly. So I'm trying to make all this stuff up as we talk. So, and human topoisomerase 2 alpha. That is one I published in peer review on Oxford University Press, no less. And this is, it's just going to be helpful to see what the spelling of a protein is. We could do this also for Nylonase. And so sequence. Let's download the sequence. We can actually see how it's spelled. And we'll do a Nylonase one. So you can see M stands for the methionine amino acids. I don't know if you could see my cursor. E for glutamic acid, V for valine, S for serine, P is for proline, L is for leucine, Q is for glutamine, P is proline, V valine, N is for asparagine, E is glutamic acid, and asparagine, and methionine, et cetera, et cetera, et cetera. And F is, down here is F is for phenylalanine. So this is the topoisomerase enzyme. About 1,000, let's see, did they list it? I don't see it here. about 1,531 amino acids long. And this is the spelling. That spelling, let me see. The shape of topoisomerase 2 alpha. So what I'm trying to emphasize is shape. The spelling dictates what shapes are possible, and sometimes it'll naturally just, if you spell it this way, even without chaperones or whatever, sometimes they'll naturally adopt this shape by itself, which is amazing. And you change the spelling, you will change the shape. That's very important. You change the shape, you change the function. So just like parts of a car, you change, a lot of it's made of metal, you change the metal shape, it has a different function. So, granted, they're different grades of metal, but I mean, you can imagine like a piston and then the engine block. They're both made of metal, but the way the machine is able to work is you shape the parts differently. There's an analogy to this even at the biological nanomolecular level. So we've gotten that out of the way. So are you familiar with, you articulated what's called the central dogma properly. And this relates to Nylonase. And I'm going to just show a diagram of the central dogma. And there are nuances to this that we don't need to get into. And I will pause a little bit and then talk about Nylonase a little more for the sake of the audience. We need to backtrack a little bit. But let me just complete this thought. So you have DNA. DNA has an alphabet of A, C, T, G. And it can be converted, transcribed is the proper word, transcribed to an RNA. RNA has a similar alphabet, A, C, U, G. And then it is then translated into a protein. So this relation between DNA and the protein is called the genetic code. I'm going to pause there for a little bit, and I'd like to just share with the audience this thing about why Nylonase proteins are such a big deal.
I'm glad you're doing that, because that's kind of why I brought it up.
Okay. So why don't I just read this? This is so good. And by the way, I'm delighted. See, this is why I hate these short format, like try to fix and learn about creation in 1 hour. This is such a difficult topic. I mean, I could teach the basics maybe in 1 hour, but then you have this deluge of just misdirection, hoaxes, lies, confusion, deliberately sown confusion. And that's actually the hardest part to deal with. The basics, learning the basics is easy. Learning how to deal with all the falsehoods, that's a gigantic task. So here's the first falsehood. This was published in 1985. And what Dr. Sanford and I worked on was demonstrating this was a hoax. So 1985, why don't I just read this? Creationists, can you see this okay?
I can.
Okay, so let me try to make this as easy to read as possible. And all right, I can't. Oh, maybe. Okay, so this is from the National Center for Science Education. The joke is NCSE. NCSE actually stands for National Center for Selling Evolution, because they're not really very scientific. They're a propaganda outfit trying to influence public policy. And this is an example of one of the hoaxes that they perpetuated. I'm glad that they continue to leave it on their website, so for all to see. because this myth has been promoted. Creationists, okay, so the title is New Proteins Without God's Help, published summer 1985. Creationists seem to be proud of their calculations that supposedly show how thermodynamics and probability prevent the transformation of biologically useful macromolecules such as enzymes. Their quote-unquote evidence usually consists of quotations from such authors as Hubert P. Yockey, who agrees that catalytically active proteins cannot occur by chance. Yockey, 1977, A and B, looking at fully evolved proteins, says that their information content is too high for their chance formation. Creationists do their own calculations to show that chance formation of biologically useful proteins is impossible. These calculations almost always involve the erroneous assumption that each of the many amino acid positions in a protein must be filled by the one particular amino acid suitable for that position. If I may pause, this is again talking about the spelling problem. Spelling, basically spelling. Since there are 20 different amino acids available for each position, the chance of randomly getting a string of 200 amino acids all in the right order is 1 divided by 20 raised to the 200th power. That's correct, by the way. If you plug this expression into a calculator, it will tell you that it equals essentially 0. Thus, the creationists say you can't get such a protein by a chance ordering of amino acids. As Duane Gish of the Institute of Creation Research put it in 1976, the time required for a single catalytically active protein molecule to arise by pure chance would be billions of times the assumed age of the Earth. But proteins, even modern, highly evolved, specialized proteins, are not built with that degree of specificity. What's more, many proteins show in their structure that they were built of smaller subunit sequences of amino acids, or they have a simple metallo-organic core that could have functioned alone as a primitive precursor of today's complex enzyme. So the creationist calculation give an answer of 0 probability because the creationists make at least 2 major errors in their assumptions. They assume the degree of specificity that has not been shown to exist in real proteins, and they insist that newly formed proteins must be as efficient as their older and highly evolved counterparts. We've been trying to explain all this to protein experts at ICR for the last 7 years. We have told them that new proteins could indeed form from the random ordering of amino acids. We have warned them that their calculations were based on faulty assumptions, and soon someone would document the natural formation of a new protein from the random association of amino acids. Now, it has happened. Not 1, but 2 new proteins have been discovered. In all probability, new proteins are forming by this process all the time. But this seems to be the first documentation of this phenomenon. The newly discovered proteins are enzymes that break down some of the byproducts during nylon manufacture. Since nylon first came into commercial production in 1940, we know that the new enzymes have formed since that time. When the enzymes were first discovered, about 1975, Kinoshita 1981, it was first thought that the new enzymes arose through modification of pre-existing enzymes that had similar function. To test this notion, the discoverers looked to see if the other enzymes in the same organism would react to antibodies made against the new enzymes. But by this criterion, the new enzymes were unique. Antibodies against them found nothing similar in which to react among the array of other enzymes in the organism. Oh, by the way, we're going to go through this piece by piece. I just want to read it in whole, and then we'll take it apart later, okay? If that's all right, Anthony?
Yes, that's fine.
Because I want the creationists to get a feel how persuasive this is. I mean, if I read this as a newcomer, I'd say, man, the creationists are just blown out of the water. And that was really the mood back in 1985. But this is 40 years later, okay? But I want you to see how powerfully they're able to argue their case. They did a good job. Right? The creationists should stop minimizing how powerful their opponent is. But they're very good at lying and telling falsehoods and maintaining it. All right? And sometimes they're honest mistakes, and their honest mistakes are also pretty powerful. Again, it was reasoned that if the new enzymes were just old enzymes with minor changes to allow digestion of nylon byproducts, they should retain at least a slight amount of activity with their original substrates. But the new enzymes had no activity on biologically derived molecules having similar chemical structures. So, by this attribute as well, the new enzymes were seen to be unique. It seemed that if the new enzymes were indeed derived from randomly ordered amino acids, they would be very inefficient compared to the usually highly evolved enzyme. Since the new enzyme would not have had billions of years of natural selection to reach a pinnacle of biological perfection. It has been shown that one of the new enzymes, the linear oligomer hydrolase, has about 2% of the efficiency demonstrated by 3 other enzymes that perform similar reactions with biologically derived substrates. Thus, by this criterion as well as others, the enzyme appears to be newly formed. More recently, another analysis, Ono 1984, I'm just going to highlight that, added further evidence that at least one of the proteins was formed from an essentially random sequence of amino acids. This evidence is a little bit more difficult to understand since its comprehension involves some understanding of how the genetic code works. I'll just have to refer readers who do not have this background to an explanation such as Suzuki 1976. It appears that the DNA that formed the gene was somewhat unusual since it could be read without finding a stop word in any of the 3 reading frames. Okay, we'll get into that. It can be shown that such DNA sequences could easily occur through the well-known process of duplication. The DNA sequence suggests that a simple frameshift mutation could have brought about the chance formation of at least this one enzyme. Frameshift mutations are known for forming totally new and essentially random arrays of amino acids, since the code is read in a new reading frame. Usually the proteins that are formed by frameshift mutations are totally useless sequences of amino acids that have no structural, antigenic, or enzymatic relationship to the original protein. This time, however, the new protein was useful. Being useful, it was retained by natural selection and was finally discovered by a biochemist who noticed a bacterium that could live on industrial waste. All of this demonstrates that Yaki, Hoyle, Rick Masing, the creationists, and others who should know better are dead wrong about the near zero probability of enzyme formation. Biologically useful macromolecules are not so information rich that they could not form spontaneously without God's help, nor is help from extraterrestrial cultures required for their formation either. With this information in hand, we can wonder how creationists can so dogmatically insist that life could not have started by natural processes right here on Earth. William Thwaites. Okay, so, all right. Honestly, that was pretty powerful when I first read it. It was unsettling. I was reading this about 25 years ago, and I was like, God, you're going to have to bring some smart people along to help us deal with it. Little did I know I'd be one of those guys who actually solved the mystery.
I am here. Send me.
Oh, I'm here. Send me. Amen.
Yeah.
And okay, just to give you an idea how hard we had to work to really utterly demolish these claims. We had one of the top genetic engineers on the planet in John C. Sanford, who, by the way, was on the other side. He was all for this. He became a Christian, then a creationist. He began to use his scientific understanding to see how all of this was false. So he'd come from the other side. He called me up one day, and this is when I was he was sending me off to graduate school to study biology. He was sending me off to learn from Joe DeWeese. You've heard me mention Joe DeWeese, Mr. Topoi Samrace. He's a associate professor of biochemistry. And so John called me up. He said, he's really upset because there was this book uh, let's see, Adam. And let me show it. So I'm going to set the drama here, and this is how this hoax has been perpetuated. And I'm sorry to be doing, I do apologize. I'm doing all this in real time, so this is kind of like a, I don't think I have time to edit all this, so we'll just have to forbear with kind of the slow progress, but I'm giving the listeners and also Anthony kind of the feel of how it is to actually do research on these topics. So, let's see. Let me expand. But by the way, what did you think of the article? I mean, did you think it was? I'm not saying you have to agree with it. Did you feel they did a compelling case? Could you at least see how it would persuade people?
Oh, certainly. Like if you were to come on to the topic and that's your presentation of it, or anything like that is your presentation of it, that might as well just be ball game for the debate in your mind. Now there's a there's a saying of sorts that I really try to live by whenever I encounter difficult topics like you did back in the day with that, which is almost anything can sound persuasive until you hear the other side. It goes something like that. That's what the saying
Proverbs
goes.
18.
Yes.
Proverbs 18. So, you can do me a favor, Anthony. Just kind of, you have access to the internet. Can you actually run StreamYard and browse simultaneously, or is that difficult? The reason I'm asking.
I haven't tried. I can try it.
Okay. Remember, we're recording. We can always restart the recording.
Yes.
The reason that's important, I would like you, or I tell you what, just for the benefit of the reader, why don't I I'll put this up on the screen and you can tell me to scroll down or something. I'll be going back and forth because I'm going to find something. I want to find something really, really powerful, because even as recently as 9 years ago, even to the present day, people are still using this hoax, and it's going into peer-reviewed scientific, mainstream, top-tier, peer-reviewed scientific journals as evidence of why proteins don't need God's help. It's amazing.
It's because it's all they have. They don't have anything else. They have to perpetuate this one.
I know. And I've been trying to get them to fix it. It's a whole nother story, but we have falsified it. We've gotten affirmation, not from the evolutionary biologists, but from the American Society of Microbiology, which is also they're probably more respected than evolutionary biologists, honestly, because they actually, yeah, they have to cure diseases, So, I'm just letting the readers know. But if you, I'm gonna put this. If you want to comment on what I'm going to, again, that article, while I desperately try to find what I'm looking for, and just kind of give me your thoughts. I can only listen with like kind of half an ear, if you know what I mean. Just tell me when I need to scroll down if you want to
If you could find the part where they said that they had no relation to any pre-existing proteins, like it wasn't a derivative of one, because I'm almost certainly that's part of the falsification that you're going to bring up here, because they are. I'm pretty sure that's part of the lie. Am I wrong on that?
We'll go into that, but what can you if you can see my screen, why don't you read what I have on the screen? That might be what you were looking for.
I'm trying to see if it is before I read the whole thing, trying to scan over it. It might be. Alright, I'll just read it anyway, and if it's not, then, we're not really at much of a loss. Alright, but proteins, even moderately highly evolved specialized proteins, are not built with that degree of specificity. What's more, many proteins show in their structure that they were built of smaller subunit sequences of amino acids, Doolittle, 1981, or that they have a simple metallo-organic core that could have functioned alone as a primitive precursor of today's complex enzyme. So, the creationist calculations give an answer of zero probability because the creationists make at least two major errors in their assumptions. They assume a degree of specificity that has not been shown to exist in real proteins, and they insist that newly formed proteins must be as efficient as their older and highly evolved counterparts. I need to kind of shimmy around this. We've been trying to explain that this to all the protein experts at ICR for the last 7 years. We have told them that new proteins could indeed form from the random ordering of amino acids. We have warned them that their calculations were based on faulty assumptions, and soon someone would document the natural formation of a new protein in the random association of amino acids. Now it has happened. Not one, but two new proteins have been discovered. In all probability, new proteins are forming by this process all the time, but this seems to be the first documentation of this phenomenon. The newly discovered proteins are, and then that's where it cuts off for me.
Okay, let me roll it down. Okay, I'm getting close to where I need to, by the way.
Okay, good. The newly discovered proteins are enzymes that break down some of the byproducts produced during nylon manufacture. Since nylon first came into commercial production in 1940, we know that the new enzymes must have formed since that time. When the enzymes were first discovered at about 1975, Kinoshita et al., 1981, I hope I said that right, it was at first thought that the new enzymes arose through the modification of pre-existing enzymes that had similar functions. And that was what I was asking about initially, to test this notion. The discoverers looked to see if the other enzymes in the same organisms would react to antibodies made against the new enzymes, but by this criterion, the new enzymes were unique. Antibodies against them found nothing similar with which to react among the array of other enzymes in the organism. And my first thought on that of like antibodies not reacting to it is like that might just be like a signal protein that's on the enzyme that's different. That might not mean that the enzyme itself is wholly different.
Okay, hang on. I'm having a little technical problem. Let me try to recover. Let me see. Hang on. I do apologize. Can you see the screen again?
I can. It never went away for me.
Okay, good. All right. I found what I needed to. Okay, so the first thing about that antibody, this is before in fact, when I read it, I'm like, what's this doing with antibodies? I didn't realize it until just now. We didn't have genome sequencing like we have today. The cost of genome sequencing, imagine buying a house for $1, and that house only 25 years ago cost $1 million. This is what's happened this is how cheap genome sequencing has been. It used to cost $1 million to sequence, you spend $1, which is amazing. So really to sequence, like say, the bacterial genome, I'm just gonna guess it's like $1,000. This would have been $1 billion investment. Imagine, 1,000 times $1 million is $1 billion. So to do what we can do now with $1,000 used to take $1 billion, and that's in today's dollars. So all they had to be able to try to determine, how different something was antibodies. They actually couldn't break apart the DNA that coded for it. They had to go directly to the protein. In today's technology, we don't actually, many times, even now, we don't really sequence the protein. It's very, 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 very expensive. You might still need antibodies to test sequences. And I'm not familiar with that. So if someone's seeing that I said something wrong, it's entirely possible. I don't work in the realm of using antibody tests, okay? So like what we do now is we'll sequence a gene and say, hey, that sequence looks like it's going to make that protein. We actually don't absolutely know that it's making that protein until we actually find the protein in the cell. So I just wanted to explain for both the viewers and also for Anthony, why are they even introducing antibodies? So when Dr. Sanford and I analyzed this, we went right to the gene sequences itself. We didn't even touch the antibodies. So if you can frame your question again, and I'll try to answer it. I had to uncoil some things first. Forgive me. So let's go ahead and let me try to answer your question.
So the main thing I was asking was the article said that it made the claim that the new Nylonase enzyme or enzymes were not derivative of any pre-existing enzyme. As that is to say, there was no pre-existing enzyme that had a similar function to it, and then it just got slightly modified by a small mutation or something like that, then to do the Nylonase activity. They said that is not what happened, and from what I remember of reading on the technical publications, some of yours and others, as best I could, was that is just inherently not true. That is just an outright false statement, that they are 100% derived from a pre-existing, I think it said like a carboxylase group, or I might have that wrong. It's been a while since I went over it.
It's even worse than that. They were never new to begin with, and some of the ones that already existed could break down the nylon.
Well, that's just majorly damning for their case.
Yeah. Okay, so this is where the American Society of not molecular, microbiology. Microbiology deals with microbes like bacteria, and I think they're eukaryotic microbes as well, like fungi, yeast. I'm sorry.
Mm-hmm.
So let me see. Let's see, let's see, get the newer version. I'm gonna, just for the reader's benefit, I'm gonna show, people will say Sal is just so out of whack, he's a creationist. Why doesn't he submit this to peer review? Well, we did, and we got rejected by the evolutionary community. But enter in, evolutionary biologists, contrary to what they claim, don't have a monopoly on biological research. In fact, modern biology is proceeding quite well without a lot of need for evolutionary biology, honestly. And so we've been rejected 7 times over 8 years. Finally, we've gotten invited by an editor to submit, and we got told we'll get a fair hearing. What drove that was this in the American Society of Microbiology. And I'm going to try to answer your point. I want you to hear the real state of affairs. So it came, their endorsement was really kind of more or less like, it wasn't the main focus of their article, but they referenced us. So they talk about plastic degrading potential across global microbiome correlates with recent pollution trends. So they're really looking for ways. Okay, nylon is considered a plastic, or close enough. Mm-hmm. And so they're interested in bacteria that can eat nylon because they want to reduce pollution. So this is a publicly available article, peer-reviewed by the American, and you can see the logo here, American Society for Microbiology. And so let's see if my name is in there. Look at that, Sanford ST and Cordova ST and Sanford J 2019, testing the hypothesis that the Nylonase NLB protein arose de novo via frameshift mutation. So they looked at our article, which was not peer reviewed, but they cited it as authoritative. And so let's look, citation 102. Okay, so he said enzymes degrading other plastic types have been shown to be widely occurring with numerous homologs in diverse organisms and likely arose from well-conserved, that is pre-existing, general enzyme classes. And so in our paper, I'm sorry, please stop me if there's something you'd like to hear, if you'd like me to explain more, or...
So,
Um,
what I'm in an effort to cut to the chase, you can continue to explain the rest of this. Sorry, I sort of kicked my desk here. In an effort to sort of cut to the chase on it, uh, was it? Did it turn out to be that the Nylonase enzyme was just a pre? You said it was pre-existing, and it was just flipped on epigenetically or something like that. Like, oh,
no,
here's the
not
present
even.
of the.
No, not even that. This is what's so bad. Okay,
Okay.
all right. So our original paper said we found that the Nile B protein, that is one class. There's several Nylonases. Okay, just need to clarify.
Mhm.
There's several. There's not only one, but there's one specifically called Nile B. We found that by the way, Nylonase is a colloquial term. You won't see it in professional literature
Okay.
as an official term. I just need to. Okay, you'll hear it by its formal term, which is kind of nasty. Like I don't even know that. I can't even repeat off the top of my head, even though I wrote on it. I mean, it's a long term. We'll just call it Nylonase, but just for your sake and others, if you Google Nylonase, you will not necessarily get all these easy hits in professional papers. Okay, from what we wrote, this is our paper. We found that the Nile B protein. Try to get this bigger. We found that the Nile B protein is widely occurring, has thousands of homologs. That means there's many of them that, if they're not exactly Nile B, they look like it. It's like, you can look at one human and then another human. They're homologous to each other. They have a similar form. Even if they have differences, they're similar. They're they're homologous. That was a
Yes.
core term. Okay. Has thousands, possibly millions I'm guessing. But it's thousands of confirmed homologs and is found in diverse organisms in diverse habitats. Likewise, we found that the other known Nylonases are also widely occurring, have thousands of homologs, and are found in diverse organisms and diverse habitats. Now look at the language that they had in this peer-reviewed paper. Similarly, enzymes degrading other plastic types have been shown to be widely occurring with numerous homologs in diverse organisms and likely arose from well-conserved general enzyme classes. They specifically cited our paper. They used essentially the same language. And importantly, they cited another paper by different group that came with independently confirmed our results. So Dr. Sanford and I were absolutely not in the minority here. We're not, people can't say that we're just cranks and we have our own ideas. We got cited, we've been agreed upon. And I also have evidence that even some hostile reviewers have said, well, you're right, Sal, but we're not going to publish your paper. I appreciated the fact they'd said it like it is. They agreed with our conclusions. They said our paper sucked, which is okay. Maybe it sucked, but we were right, right? You could say the right thing, and maybe the way you say it sucks. I don't think so personally, but I didn't care. They said our conclusions were right.
Well,
So
who
this
was
is
saying that?
so bad, huh?
Who was saying that the paper sucked, but that you were right?
I have it. I have it on record. I can find it for those interested, even if we want to do it right now. I think that would be helpful. Why don't we do that?
I'm
This
down.
is, all right. This is by cell biologist. He's the editor-in-chief of Cell Biology Report of Cell Reports. It's a peer-reviewed secular editor-in-chief named Steve Matheson. He was on the Peaceful Science website run by Joshua Swamidas, a theistic evolution website. But
It's
really,
biologos, right? Or is that
he used
is
to
his
be part of.
associate?
He used to be working with Biologos.
Okay.
So, going back to that 1985 paper, they're saying this is unique. It never existed. It's brand new. So the first thing is they didn't have all the bioinformatic databases. Remember, it's a million times cheaper to sequence genomes, which you can buy for $1,000 would have cost $1 billion. So there's no way they could know the level of what we know today. And what we know today is that these were not unique. They're all over the planet. So
That
someone could
was
say,
Well, I was going to say that was going to be my next thing I was going to ask about you. Is my volume fine? I noticed you were trying to hear me.
No,
Is it better now?
we were talking over each other because of the
Oh,
lag.
okay. Okay,
Go
okay.
ahead.
In your paper and how they cited you, it said that diverse organisms and in diverse environments, and the immediate question that popped into my head is like, okay, is that enzyme present in organisms that aren't actively using it in places where there's not nylon byproducts to consume, or not enough to matter? I would say, no selective pressure
Okay.
there.
The reason. All right. So something that. Okay. Let me just give an analogy. I could take a screwdriver, right? I could use it like a hammer.
Yes.
It turned out nylonases, what we call as nylonases, they actually have an original function. They can break certain, what they call amidase bonds or peptide bonds. Or okay, a peptide bond is an amide bond, but an amide bond isn't necessarily a peptide bond. Just for you aficionados out there, not necessarily just for you, Anthony.
So I know what a peptide bond is. Would you mind defining an amide bond?
You'd have to ask a professional chemist. They know
Oh,
that.
okay.
I do appreciate it. So I'm going to give. Oh my goodness. This is it. I found it. You can, okay, the Googlers out there can find this interaction. And they were ripping on me. I got to tell you, Dr. Sanford read what they were saying, and Joe DeWeese read it. Joe DeWeese was a co-author, although not listed. He should be in our next iteration. Remember, Dr. Sanford is a famous geneticist. Dr. DeWeese was appointed by the governor of Tennessee to set science standards. And he's a professor of biochemistry. And they're just ripping on us. And they were horrified at the comments. Okay, so I'm not saying that I agree with their assessment. The important thing that I wanted to get across, it's like, all right, say we're stupid, we're incompetent, but we are right. Okay, so let me show you this. And Steve Matheson is the editor-in-chief of Cell. And so for those out there who want to find this, Google Cordova and Sanford good enough for a secular journal. And the website for them it's on is Peaceful Science. And here they're ripping on us. What a lousy piece of garbage we wrote. And so it's vindicating that we got cited by the American Society of Microbiology as authoritative. So I'm just trying to just show, don't always believe people that are that biased that we were, not worth listening to. The bottom line is, you'll see it when Steve Matheson weighs in. So they're ripping on us and like, you don't know what you're doing. And actually, I got to tell you, this was actually beneficial because their criticism got me to fix one minor thing. They didn't like the way that I used something called SciBlast in my search tool. I said, dude, what I did is I redid my database searches with the way they suggested. Now they don't have anything to complain about. What excuse do they have? So they actually gave me free of charge editorial review, probably worth thousands of dollars. So thank you. It was worth the insults. Because now it became a better paper. What the American Society of Microbiology did was they cited the second version of our paper. And so they've actually helped it. So you have all these people. There's a back and forth here. And I'll cite the guy. Oh, here it is, Steve Matheson. Steve Matheson is the gentleman's name. Here's the important part. Stephen Matheson. And you can Google him. He's the editor-in-chief of Cell Reports, which is very prestigious. The parent one is prestigious. Cell Reports is kind of is a notch down. He says, "Oh no, was wrong. Oh no, was wrong." That little thing he said, "Oh no, was wrong." This would falsify everything in that Proteins Without God's Help. He didn't elaborate, and I can understand when you want to admit that your side had made a big blunder. You don't want to emphasize. You don't want to highlight your side's mistakes. So he just simply said, "Ono was wrong." And if you go back to that article, Proteins Without God's Help, they were citing Ono as authoritative. Matheson said Ono was wrong. And this also destroyed Dennis Venema's book. He wrote an entire book. He used that example. Venema used that example of this hoax, as his favorite example of evolution. He wrote an entire book. He said, "This is my favorite example." And I could quote this. So if you have any thoughts, what do you think so far?
I mean, it's right in line with everything I had sort of read and researched up to on my own to this point. Obviously, I don't have the in-depth understanding of it that you do because, well, that's why I'm here to learn from you. And you're kind of one of the guys, capital letters, who really took it down. So that's kind of why this is one of the best places to learn about it from you. And my thoughts on it is just like how you were saying, he wrote an entire book and then cited that as his favorite example of evolution. It's like I sort of weighed in earlier. It's all they've got. It's Nylonase and Richard Lenski's long-term evolution experiment with CIT+, and like that's it for like new. There's a handful of smaller, much more ambiguous other ones, but like as far as there are any like sort of heavy hitters that actually have a real chance of being like, oh look, new genes can actually arise through random chance, functionally. That's pretty much it. And CIT+ was pretty much done away with pretty quick because they're just like, oh, well, this is just an instance of a duplication messing up a promoter region and turning the gene on all the time when it was normally there but only supposed to be on in anaerobic environments. So it's like, yeah, that's not a new gene. That's just a ceasing of functioning of regulatory systems. And with you here with the Nylonase, they were really excited about that because they're like, oh, it definitely has to be novel because we only started producing nylon in the mid-1900s, and so it definitely can't be something that pre-existed stuff. But then once we get the gene sequencing technology to figure out what it actually is, we start sequencing a bunch of stuff that doesn't come from a factory wash-off pond full of nylon byproducts that it could dissolve. It's like, oh, why is this thing over here in South America have this gene? Or, I don't actually know the different locations that were cited from, that would have gotten any sort of samples to have sequenced the Nylonase, or NILB, whatever the official terms are, out of it. But I hope my point is getting across.
Oh yes, I am sorry I'm fumbling through with my explanations. It's partly because I'm scrambling to, it's powerful when you can actually see the citations, not me just saying it, if you know what I mean.
Oh yeah, because then they can't just say, oh, you're lying. It's like, it's right there. It's right there. Look at it.
Exactly. And let me see if I can download my supplementary materials. Maybe I'll have something I could actually show.
But yeah, that's why they are. Sorry, the lag kind of got in the way. I didn't realize you were talking.
No, go ahead.
Yeah, but that's my thoughts on why somebody would basically cite Nylonase as their favorite evolutionary example, even though it's dead wrong, because it's just like, you have to. It's all you have. If it's all you have, you got to run with it. Otherwise, your theory's bankrupt, right? I mean, maybe you can sit back and say, okay, well, we haven't, which is probably what they're going to start doing now, because to my understanding, I haven't seen too many people in recent times really hop on the train of using Nylonase, but what they'll do is they'll sit back and they'll go, okay, well, maybe that one wasn't an instance of actual de novo functional proteins appearing or enzymes appearing, what have you, but it's just like, but we'll find it. It'll happen. We'll document it one day, and then you'll see. And it's like, and you're allowed to do that. You're allowed to do that for your theory. You're allowed to sit there and make what's called an appeal to a hypothetical future vindication. Almost all theories do that. But what you can't do is say that the evidence is on your side while that's all you can do. You cannot sit there and say, oh, this is such a proven theory. This is, scientifically airtight. There's no way you can argue against it, when what you're having to do is, when your objection says, actually, this has never been shown to occur, and this is not minimally, this is majorly necessary for your theory to be true, and they go, oh, well, we just haven't found it yet. It's like, well, how supported of a theory can that really be? If something's so central, something that you haven't ever observed, and even the examples you try to cite of it eventually turn out to be wrong. They get vindicated in the wrong direction for your stuff to be right.
All right, so since you asked, you asked about amidases, and I'm not going to answer this completely because, again, if you'll excuse me, that it is outside of my expertise. So what do we do in such situations? We find someone like Joe DeWeese who can kind of bail us out, and so we hang on, I'm trying to find my StreamYard. So I'm going to, just for the reader's benefit, because it's like, okay, I was in a graduate level bioinformatics class. We had all these postdoctoral student fellows. That means guys who already have their PhDs or MDs. Classroom fellows, smart guys. And they bought the line that, oh, you can't have an enzyme that can break down nylonase, since nylon wasn't invented until 1935. And I'm like, okay, so a rock has the capability to break a window. Did it suddenly evolve the ability to break a window? Could it only evolve that after windows were made? I'm just like, that's ridiculous. Does that make sense? It had the inherent ability to break a window even before windows were first made. But they're so in the evolutionary mindset, it's like, no, there's no way an enzyme could pre-exist that could break down nylon until nylon was invented. It had to evolve. And I said, look, did rocks suddenly evolve the capability to break windows? Did they have to wait till windows were first invented for them to develop that? You have such the evolutionary mindset, you can't even see that possibility. And I'll show you why it's not. It's helpful to understand what the purpose of an amidase is and why it can break down nylon oligomers. And I use the word oligomers. So when you have a polymer, it has all these little units like spelling, little, just like beads on a string. A polymer is generally something that is really long, but not always. I mean, depending on who you ask, okay, and what convention is being used. An oligomer, you might say, is also a polymer, but it's short, something like less than 20 units. The reason nylon-eating bacteria really doesn't eat nylon, if that were the case, people that are use parachutes, they'd have to be worried about nylon-eating bacteria eating their parachute. Why doesn't that happen? It's because the nylon in a parachute is a very, very long polymer. What the bacteria were eating were small little segments, short, say around less than 20. Many of them were only like 3 or 4. There was a table we actually studied, said, the number 12 was about really how big. So these aren't even real nylons. They're not commercial grade nylons, okay? They're waste products. These are disposed of. Yeah, go on.
So is it, at that point, is it chemically the same thing as nylon in terms of like the molecular structure, and it's just not as long? Is that what you're saying?
Yes, and that's important because I'm showing this diagram here. I'm going to show what a human, you're going to see what a human, all right. In a human, remember like the topoisomerase or any protein, it will have this structure here, and the chemists will recognize this. This is the amide bond, and it's a peptide bond, but it's an amide bond, meaning, there are many kinds of amide bonds, and one subset of amide bonds is peptide bonds.
Okay.
And I want the readers, even if you're not familiar with organic chemistry, the O is for oxygen, H is for hydrogen, N is for nitrogen. So you have hydrogen, nitrogen, oxygen, and they're positioned in this particular way. I want you to see how we highlighted that in green. Now you can,
I think the actual image went away. Like I saw you pull it up, and then after a moment it went away. So if you're trying to show me something on the screen, I'm not seeing it.
my bad. There you go. All right, let's
All right,
start again. Apologies.
that's
This
fine.
here is a fragment of a representation of a human protein. And even if you're not familiar with skeleton diagrams and organic chemistry, I'll at least read you. Okay, the diagram is supposed to show the atoms in relative position chemically. So, what connects when you saw a protein and how the amino acids are hooked together? They're hooked together by a bond called a peptide bond. And I'm highlighting it in green here. Here's a peptide bond right there.
Okay.
So one way you could see it is you have O for oxygen, H is for hydrogen, N is for nitrogen. Now you could also flip it upside down. It's still the same bond. O is for oxygen, N for nitrogen, H for hydrogen. So this is a peptide bond, which is also an amide bond. This is a peptide bond, also amide bond. This is in biological systems. Anything that has proteins will have these bonds. Now it turns out there are enzymes. When you're eating food, you'd like to be able to break apart the proteins, right? And you reassemble it. So when you eat meat, you may break apart the protein into amino acids, and then you rebuild it into the spelling that suits you. That's why you can eat proteins from a cow, and even though their proteins are spelled slightly differently than ours, when it integrates the amino acids from the cow integrate into your body, the amino acids adopt, the proteins in your body adopt the spelling of the amino acids that are unique to humans. So there are plenty of enzymes that will break this peptide amide bond. Now, if you look at nylon, doesn't that look like a similar bond? The enzyme just has to break this. It's the same thing. And I showed that to Joe DeWeese, who's a biochemist. He said, oh yeah, that totally makes sense.
Okay. It's all coming together. I'm putting the puzzle pieces together now. I see why it's such a. Every time I kept asking you about the, oh, is it this, and that's why it was bad for him. You're like, no, it's even worse. I get it now. It's literally just the enzymes to break apart the bonds that nylon consists of have always been there, and they just started doing that when nylon was produced because nylon requires those bonds, and the enzymes can break them.
Yes, yes, you get it. You totally get it. I couldn't get these postdocs to understand that. I mean, it was unbelievably frustrating that look, you're PhDs, you're postdoctoral fellows, you're at the NIH, you must have had biochemistry, and you're believing stupid stuff like this because the evolutionary biologist told you so. And I knew I'd won the argument, not only later because of Steve Matheson, but my professor was a top-tier evolutionary biologist, by the way. I presented this to him. He said, you're right. Ono was wrong. Didn't take him 2 minutes. Didn't take him 2 minutes. And yet Dennis Venema wrote this book. Dennis Venema is a professor of biology, proponent of evolution. He wrote an entire book. He said, this is my favorite example. And worse, Ken Miller, who was in the Kitzmiller versus Dover trial, arguing for the Darwin side. And by the way, the Darwin side won that case. That's another story. Ken Miller, this is one of his favorite examples. And he's a professional professor of biology. The kind of junk that these guys that are supposedly top tier defenders of evolutionary biology, they're promoting stuff that my professor of evolutionary biology saw it was flawed in 2 minutes. All I had to show him was I did some sequence comparisons between, just like I showed that all these widely occurring habitats. Mm-hmm. It's very easy now to do in the era of genome sequencing. Just put in a search term, say blast, and you get 20,000 hits, and you realize they're all over the place. He said no was wrong.
And the BLAST is the basic local alignment. I forget the last two of it, but it's basically a software to do local alignments with various gene sequences.
String search, string
Yep.
comparison. It's like a Google search. Very
Yeah,
similar.
you enter a query and it goes through the sequence and it says, okay, we have a match here, we have a match here, and you can have it like on percent matches, right? Where it's like this one would match it 80%, or this one would match it
Yes,
90%. Yeah.
we were getting. It was so unbelievable. In fact, there were so many. I was like, there's a cutoff to how many matches you'll get because once it starts to get about beyond like 30, 40, 60,000, Blast will just quit and say, look, dude, this is a kind of pointless. You have so many similar matches. Where do you want to, can you kind of like improve your search? So there were so many similar matches. This is again, because we can equate the spelling of DNA and proteins, the English alphabetic characters, you can totally see how things like search engines can almost do the job for you. Now, this BLAST is specialized toward this, okay? But it's kind of the same thing. I mean, anyone can kind of say, look, this is plagiarized because the text is almost identical. It's kind of the same thing. And so this is how we're able to establish it. And then, so, okay, how long did this take us? We had Joe DeWeese, John Sanford, myself. I worked on this project for a year. A year. So when you saw that presentation by the NCSC in 1985, that was powerful. I said, John, we're going up against Goliath. Of course, we know what happened to Goliath. And I said, the first round was not very promising when we first started. I said, John, we barely have a case. I just did a blast search. And then I refined it to do proteins and like, oh my goodness. Even then I felt weak. And then I went to Joe. I said, hey, we still have some mysteries to solve. Why does we have to make the case that this is used for something in biology. And we found out that diagram we just looked at with the peptide bonds. Now we could see why it pre-existed. And if I could rub it in just a little more. By the way, what I've told you is still not the whole story. So you're going to hear some things that are like, what about this or that? And I'm just like, look, there are so many nuances to this. It could take days to cover it, but I've given you the essentials of the story. So when you're going to hear, what about this protein and this experiment? There's so many of this, but I gave you the kind of the essentials. So let me, I'm trying to get Dr. our original paper because I had some other things I wanted to say. In the meantime, do you have something that you'd like to comment on?
I'm just really glad that you clarified that for me because it definitely brings a lot of the technical papers I've read from you and other people. It brings it all together. I'm like, oh, okay, because my limited understanding of the various higher, deeper weeds of the technical field does limit my ability to comprehend things in the papers. I think I do pretty good. I do as best as I can, and I get it pretty good. Yeah, but it was like a eureka moment once you showed me that diagram of it. Be like, hey, this is the bonds in nylon. This is the bonds that these pre-existing enzymes break down already. They're the same bonds. So when the nylon is a short enough polymer or oligomer for the enzymes to get their mouths around, they can break it down because they already know how to break those bonds down. And it's just like,
Yep. Okay. You see, this is why I'm very passionate. We can win this fight if people are willing to spend time like you to learn. Otherwise, if they just keep going at a level that is partly because the surface level, or also because even if the data is out there, we're writing in such technical terms for for people in our field. People outside the field will not understand that we actually won this case. There's still people that are saying we're stupid. I'm just like, no, we're not, and we're right on top of that. And it's only when people actually study the matter on purely technical terms, in light of the data we have, they'll start to see it. But this is the first time I've had the chance to actually walk someone through who's outside the field. They can see it's not that hard. Look, it didn't even take us 2 hours, and now you have more comprehension than these PhD postdocs who are my classmates in bioinformatics. They couldn't get it. They refused to. They're willfully blind. They sort of resented that I asserted that. I think they hated me. They were sneering and practically, which is kind of rude, but that's their attitude.
Well, you basically just showed them that their New Age Piltdown Man was a hoax. I don't know if you remember what Piltdown Man is, but it's a pretty classical example of an evolutionary hoax where it was literally a orangutan's jaw glued to the bottom of a human cranium. Like, they took the actual mandible off a human cranium and glued an orangutan's one. They filed the teeth to match. And for, I don't remember how long it was, but it was years where anthropologists and evolutionary biologists and paleontologists all said, yes, this is a crucial missing link, and it was literally an orangutan's jaw glued onto a human head. And it's like, my man, that's bad. And that's, this is definitely a lot more complex of a thing to get into than that is, but, it's kind of an equivalence. Like, they're leaning on this as a really big deal, and when you come and show them that, hey, your big deal that really proves your case is just outright false, they probably can't accept that, because it's like, well, it's not just that they lean on it heavy, it's one of the only things they can lean on, and if they don't have it, they're going to fall over.
I'm bringing. Okay, so this was okay. Let me back up a little bit. So this was the paper, "Birth of a Unique Enzyme," that they claimed this was brand new. And if you look at cited by other articles, this is really funny. You could see some of them. You could see some of them as recent as 2025 still citing it as authoritative. I mean, this is hilarious. And I'm like, okay, you want me to... Okay, you have books being written by like people like Dennis Venema, but then you have people, okay, look at this. Nature Genetics, which is a prestigious journal, Nature Genetics. And they cite Ono's nonsense about Nylonase as an example of proof. New genes is a general evolutionary process. So this garbage is continued to be recycled. We got blocked from publication. The editor said, well, go find another editor. We agree with your conclusions, but they gave a whole bunch of excuses. They said, well, good luck. You're right. Good luck. We won't publish. You can find someone who'd be much more eager to publish. I'm like, okay, you're at least honest enough to admit we were right. And there was one case, there was an editor, we actually liked him, and we could tell he was afraid for his job. And John and I actually had Dr. Sanford, that is, John Sanford. Mm-hmm. He insists I call him John in person. So sometimes I'll slip into that. So John and I said, he seems like a decent guy. He kept encouraging us to submit on every other topic except this one. He said, please come back and submit again to this journal. But you could sense he's like, I do not want to publish on this topic. Talk about something else. You're getting me in trouble. And I said, John, I think you and I are kind of seeing the same thing. He's a good guy. We're grateful, but he's afraid for his job. And we actually didn't want to imperil him. The reason is there are now people that have jumped ship on an individual level, and they're in a crisis. They're like, I'm now an editor of a journal, or now I'm in a prestigious position. I no longer believe in evolution. What do I do? And sometimes John and I have said, look, this is not the time for you to come out of the closet. Let your career grow. When you're close to retirement or you're so well established, that's the time. We appreciate that you're willing to sacrifice your career today, but you're more valuable to us, to the movement, by staying there, keeping a low profile. God will give you the time when it's time for you to stand up. And if God calls you to sacrifice yourself, then do it. He knows your heart that you're willing to do this today. But we're counseling you just to wait. Your time will come. Um, you'll have plenty of opportunity to have yourself sacrificed and die in battle. This is not today. And so I'm not trying to say all the editors were bad guys or bad actors, but there's just this general hostility that's making it a terrible climate. And so did I show you this one here? Let me just make sure people see it. This is citing that garbage example. This is like the modern day Piltdown man still being used as evidence. Still being used as evidence. They're citing Ono 1984.
What year was this published? This one that you're showing me?
2025.
That was published this year. I mean, we're at the end of this year, but that was published this year, and they're still doing it.
They're still doing it.
Like I said, I'm sounding like a broken record. It's all they got.
That's all they got. And I'm just laughing. Yeah. People say, why don't you publish in peer review? I'm going to point to that examples like this. I said, because guys like you, guys like you who keep taunting us that way, you'll be the same sort of peer reviewers that'll let garbage through. And you're not calling it out. Why don't you clean up your own act? Why don't you publish something even you knew, like Steve Matheson is an editor of Cell? You know that this is wrong. Why don't you do something about it and set the record straight on, except just maybe, he'll say, I did say he was wrong on some website. It's like, you'll clear your conscience that way. You'll actually not try to go through the process and see how hard it is to get the record straight. Thankfully, an editor finally came out and invited us to set the record straight, but this is after 8 years. And in the meantime, all this garbage is continuing to be recycled. I'm sorry to get on my soapbox. You touched on a topic dear to my heart, because this has been a long-standing battle, and I've gotten to see kind of the ugliness of the whole industry of evolutionary biology in the process, how corrupt it is, and even for the honest people there, how they can be just either bullied or deluded and to not see clearly because they just believe their colleagues are right. Yeah, I mean, sorry.
It's a shame of it because, there's no end to hearing the tout that it's just like, 99% of scientists or whatever agree with evolution. It's just like, no, most of them are silent because they don't want to become a pariah for the academic in-group bias that'll throw them under the bus. You basically got to be a James Tour before you can really have a loud voice. They don't like James because he's speaking against something that's near and dear to their heart, which is the origin of life, and he's just calling out all of its bad premises, assumptions, and outright fabricated claims in a lot of cases. And they'll poo-poo him all day, but it's like they can't really touch his career too bad because he's so necessary. But that's basically what it takes if you're going to be that outspoken.
Exactly. And so thankfully the situation is slowly changing. Even in the last, I've talked to my colleagues, they said, I said, man, things have changed in the last 20 years. And they said, even in the last 5, because the data is flooding in and you're getting a lot of people that have the right level of expertise. You're getting people, not quite the caliber of James Tour, but people that are still pretty good. They're professors. They're long established scientists. They're kind of slowly like, we kind of see what you're seeing.
Accredited professionals across various scientific disciplines going, hey, something's not right here.
Well, because I've watched this, I mean, I'm giving away my age here, but I've watched this battle unfold over 45 years. And so it was heartening to see that article by Dwayne, that thing, Proteins Without God's Help, because that was 5 years into my monitoring of this debate. And it's like we had nothing. It looks like we're losing every battle that we tried. It's changed now. It's not that the creationist side has done better. It's that God has provided all this data, and the data is speaking for. If we remain silent, the rocks will cry out. Is almost kind of figuratively happening now. And I did want to share one other thing that you should laugh about. It's a highlight, and thank you for interest in our paper. God willing, this is going to get published in peer-reviewed, and it'll be funny even after if we do get it published, the people will keep recycling the falsehoods.
Oh, they're gonna.
So, oh, sorry. Let me get this back up. So this is from our paper. No, wrong.
Yeah,
Sorry.
I'm seeing the original article that was saying and now I'm seeing DNA, the central dogma, DNA, RNA, protein.
Yeah, that was the wrong one. Sorry, I thought I had. Oh, by the way, just before I take this one down, there's this thing called aminocaproic acid. That's basically nylon. Those
Really?
are the elements of nylon. And guess what? They prescribe it to humans. And I wanted to say, remember that peptide bond that is broken up? We have something called trypsin in our bodies. Pigs have trypsin. Mammals have trypsin. They can break down nylon oligomers. And those oligomers can be digested. Now, I wouldn't recommend you go out and do this, but there is a FDA-approved treatment for bleeding called aminocaproic acid, which are basically nylon monomers. I'm just saying, guys, it's not like it's not like we had absolutely zero that this was something so brand new.
So a nylon monomer
after 1935.
A nylon monomer would just be one molecule of it, correct?
Oh, go on.
I was asking I just wanted to confirm that a nylon monomer is just one molecule of it, right? Like it's not a chain.
Right.
Okay.
Right. And it looks very much like the lysine molecule. It looks a lot like a lysine. A lysine is an amino acid. It just has a slightly different structure. So, I'm sorry, I'm hesitating here because I'm trying to find something that we wrote, and I might have to bring it up again. I do apologize for being such a mess here, but we're trying to do this on the fly. Let me. Can you get me a chance to reset? I would welcome your thoughts while I try to get the next thing up.
Okay. So, one thing that I did also just... I just heard my computer say something unplugged. Can you hear me?
I can hear you. We're still connected.
Alright, I was hoping it wasn't my mic. My computer does a little boop whenever something unplugs, and it just said something unplugged. I don't know what it was. But, so what seems like the major issue is like, okay, well, why is nylon normally so resistant to bacteria? And it's like, well, it's kind of the same thing as why asbestos is so bad for you. The actual enzymes can't get their mouths around. They can't get properly around the long chains of nylon. But when they're the oligomers or the monomers, small enough for the enzymes to get around, they have no issues breaking those bonds down.
Exactly. So this is where we call this in the field of rhetoric fallacies, equivocation. You're equivocating something really tiny with something that's really long, and you're still saying, well, that's nylon, but it's like, well, it's exactly as you said. They are both nylon, but there's a difference in the size, and what nylon-eating bacteria can eat are not the long polymers in commercial grade. I'll call it commercial grade nylons. So like your stockings, your parachutes, or anything else that we would probably sell in the store is going to be made of really long stuff. And so the bacteria were actually eating and thriving in waste pools of basically the bad nylon, the trash.
Wasn't long enough to be useful, yeah. And because it wasn't long enough to be useful, they eat it. They can get their mouths around it, so they eat it. Mouths, they don't have mouths, but
exactly.
Yeah.
Oh, so I have here. Okay, so let me see. So what we did even I had moved on, and even my own paper, I had, as I read it, I was like, I've totally forgotten what we wrote because we'd moved. The way I learn new things is I forget old things.
You have to. There's so much, especially in the realm of like, my God, microbiology or not microbiology and molecular biology, pretty much all of it. It's like, man, there's only so much you can retain at one time, and after a while, you're going to be like, okay, even though I went over this, even though I wrote a paper on this, let me dig something back up and refresh my memory, just because there's such a volume.
I totally. I mean, I tell you, it's shocking when people ask me about this. I'm like, what did I say? You're asking me like what I said yesterday, and I was like, I feel so embarrassed. I actually had to go back. I mean, recently Rob Stadler was asking me about this, and I'm like, I totally forgot what we researched it and just moved on. But this is exactly what happened. But let me show you this passage. I think it'll be funny. In 1992, and I'm just pointing this out, someone had actually agreed with our findings. Okay, so Dennis Venema was perpetuating this myth in 2016. But as early as 1992, and we found this is in our paper, Salvador Cordova and John Sanford. We found a paper in 1992 in response to Kanagawa's discovery of P-Nalb. Yomo et al. Co-authored a paper with Urabi and Okada to put forward a third competing hypothesis regarding the origin of Nalb. It actually is the right one. So there are actually three hypotheses. Frame shift is only one of them. That's the one that's Dennis Venema's favorite. It's the frame shift.
It's also wrong.
Right. It's also wrong. It's his favorite one. It's wrong. This is the correct one. Even if you're in an evolutionary, assuming millions of years, okay, people know I'm a young Earth creationist, but let's assume we would say this is the more correct one. Yomo et al. Argued that Kinoshita's F-Nalb and Kanagawa's P-Nalb homologs descended from a common ancestor that existed about 140 million years ago.
I might have read this paper that they're referring to here. It was just so long ago that I don't remember it. Like, I don't remember everything about it, but the names are ringing a bell.
Okay, so this is important for the evolutionary biologists, not so much for our purposes. But when you have two similar, it's just like when you have Ancestry.com, you can get two random people and they could sequence their genomes and compare them and say, well, okay, you're either twins or you're siblings or you're cousins, and you had a common ancestor this far back. So the degree of dissimilar, you want it to be similar enough that you can compare them, but dissimilar enough that you can kind of estimate how far back the common ancestor is. So even under evolutionary assumptions, that Nylonases existed 140 million years ago.
And it's because of all the homologs all over the place.
Right. That's the significance of it. And then this specifically, I'm going to let me read the rest of this, what Yomo said. It's really funny. I mean, I'm just trying, I know we're kind of beating a dead horse at this point. I am having a little bit of fun beating a dead horse because I think it needs to be beaten a little more because people are still citing this.
Well, nobody knows the horse is dead.
Yeah, no one knows the horse is dead. Okay, so
they're trying to sign it up for the horse races.
That's right. So okay, we summarized it as being 140 million years ago. We'll quote specifically exactly what he said. The distance between P-Nalb and F-Nalb, or F-Nalb prime, is much larger than between F-Nalb and F-Nalb prime. The time divergence of F-Nalb and P-Nalb is estimated to be at least 1.4 times 10 to the 8th years, which is 140 million years. Therefore, most of the amino acid substitutions from the ancestor of Nalb gene family to its descendants of today might have occurred before the beginning of nylon manufacture.
Which would mean it's not a post-nylon de novo enzyme.
No. And there's no reason to believe it. That's why Steve Matheson of Cell Reports said, "Oh no" was wrong. He basically affirmed our conclusions. Your paper sucks. We're not going to publish it, but you were right. So thank you very much. And that is the Saga. And to this day, I'll still get flack from evolutionary biologists said that you have zero scientific content in what you wrote. I'm like, yeah, say whatever you want, dude. We know we can go to sleep at night knowing we're right.
Well, earlier, you were talking about how, when you first read it, it's like you're up against Goliath and, they have so much there, and then the conversation proceeded about the fact that even now in the last 20 or even 5 years, a lot of people are turning around just because the data flowing in, and it's just like, yeah, there might not be many of us young Earth creationists, but we don't have to have all the funding. It would be nice. We don't have to have all the funding and all the personnel to show the truth, because if you're on the correct side, it's going to come out eventually. Yeah. You can have your proverbial mountains of Philistines and your proverbial Goliath on the other side, but if you're arguing for lies, your time is, you're on a timer, bud. It's coming. Yeah.
And this is at a very deeply, and you could tell how passionate and emotional I was getting. Imagine me going through 40 years of this and thinking, man, there's no way anyone could fight this unless there's some bright guy who's going to come along and dispute it. And like, John Sanford, oh, you're the guy. Like, why me? I hardly know anybody. Seriously, I hardly knew any biology when I started this project. This is part of this was my first real project that I worked on.
What a way to kick things off.
So I had basically about your level of where you are now. This is, just imagine, of course I had help of really two senior guys. So this is the other thing is, if you can get mentored by the right people, you can do great things. It's just drawing out the potential. But if I had to start on my own, there's just like no way I would have been able to do this. So I glory and honor to our Lord and Savior Jesus Christ for, providing for us.
That was definitely some divine providence going on.
Amen.
There.
Yes. So we're at the 137 mark. I've had a lot of fun. I feel bad kind of have done all the talking. I mean, so maybe we can start thinking about other things we might want to talk about. You don't have to tell me now, but before we, I'll meet you backstage. I'm going to end the recording. We'll have a little bit more conversation backstage. But do you want to say anything to the audience? Any thoughts about what you heard today, or anything?
I'm just formulating it real quick so I don't trip over myself near as much. To the audience, I hope anybody watching this can really learn that just because the evolutionary story comes out with some new thing that they've proposed and it seems insurmountable, it seems like a Goliath, there's almost always not near as much weight to it as they're presenting it at first. As a matter of fact, I can't really think of an example where even things that are still slightly in their favor don't have near the weight they put on it. And earlier I mentioned something called an appeal to hypothetical future vindications, and that every theory and every idea does that. And one of the reasons that I ended up being a young Earth creationist is because the future vindications have almost always been on the side of the young Earth creationist stuff. Like, we have the Nylonase that we were discussing here, that this was this whole video. And another one that comes right to my head is like the Y chromosome and mitochondrial sequences that trace back to, it gives a roughly 6,000 year and roughly 4,500 year for the Y chromosome and then mitochondrial. And it's just like, that's right on the money for, the biblical account of our origins. Now, there's some debate within the idea of like, can those things be a reliable molecular clock? And, there's some discussion to be had there, but it's like, even still, the fact that it gets anywhere near it is very damning to the opposition and very vindicating for our side. And there's just a whole lot of stuff like that on almost every single topic. What was it? Was it the NIH, the National Institute of Health, that did that figured out that the human genome was almost they said it's at least 80% functional, and it's like,
The
well,
encode
now,
cycle.
yeah, and it's like, well, now you guys have a bit, your junk DNA myth is gone. You've been leaning on that for however long, decades upon decades, not you, but the evolutionists, and it's like, y'all needed that. Mutational load is just going to throw your theory out of the water now. Now, your PR as an evolutionist is the best it can be. You have all the institutions and all the governments on your side, so you're going to be able to trick people for a while, and especially anybody who doesn't listen to it or doesn't, look into it deeply. But it's just like, you're already wrong now because of that. The facts are not on your side. You're just playing a PR game now.
Well, okay, so with mitochondrial Eve and ENCODE, I'd say they are good arguments. I wouldn't necessarily yet put them at absolute top tier, and maybe that would be a good point of discussion, since, as I mentioned earlier, I'm trying to write a booklet, 20 pages, and I'm going to just give the top tier of arguments.
I could agree with that. That's why I said that there's some back and forth on, if those molecular clocks are as precise.
And how do I judge what top tier arguments are? It's when evolutionary biologists themselves had admitted it. And a lot of people don't notice. So among themselves, they'll publish, and it's not a secret. They can clear their conscience, and I salute them for that. But the popular science writers are not going to jump all over it. And even within the community, it's not going to get the most attention and grants and money because it's like kind of uncomfortable. But there have been top tier people that have said this, and I'm like saying, this is what I was saying even in the last 5 years. We might stretch it to 20 years back. I started to see stuff. I'm just like, I don't even need to appeal to me and my knowledge or make arguments. They have made it for me. So if you'd be interested in going through my perspective book, The Elements, you would be a good person that I could kind of run this by and say, did this make sense? Speaking of which, I hope I made this presentation, it was technical. I hope I made sense, that you could see things more clearly.
You brought a whole new level of clarity to the Nylonase issue. Like I had a grasp on it, but there were just some details that I just was fuzzy on just because of my ability to cut through the technical weeds wasn't as great. But then I have someone like you come in here and explain it to me, and the light bulb went off. And I'd be more than happy to assess your book or booklet and be like, hey, this was pretty easy to comprehend, or this was a little hard to understand, made my eyeballs feel hairy.
Okay, great. Well, no, this is a godsend, because I've tried to tell people, just give me a little more time than 10 minutes, and I can give me more time, like say, just like today. It's approaching 2 hours, but it's not. You got clarity. What ends up happening is creationists will sometimes put these really lame arguments, and they spend days on the internet trying to defend it. And like, look, you could have done this more efficiently if you just chose better ground to fight on. They say, pick your battles, choose your battles. And sometimes people are dying on a hill that they don't need to. They want to do the human chimp ancestry. So we could talk about things not to do, and what I would think, if I were trying to teach someone, what would be the best arguments. The best arguments I can give, and I'm just going to give you a sneak preview. I've been in an ongoing battle with various evolutionary biologists on genetic entropy, and I finally found the solution. I found a top-tier evolutionary biologist who's pleading for intelligent design to save the human genome. He said, well, okay, I don't think he believes in God or in the intelligent designer, or even if he does, he still believes in evolution. But his name is Alexei Kondrashov. He wrote the book Crumbling Genome. He's a geneticist, studied human genetics. Now, if you study human genetics, you can't help but notice there are a lot of heritable diseases, and they could be actually increasing. Again, in the era of genome sequencing, we might actually be able to see this in real time. And he also knows about mutational load, and he's very disturbed by it. So I was in a debate 5 years ago with another evolutionary biologist, Daniel Stern Cardinal, and he was hammering on me, trying to hit me hard. And so, the first 2 hour. It was like a equivalent of a 4 hour debate, was split into two debates. The first 2 hours he was hammering on me pretty good. Um, Again, because it's like I hate these short debates. They can use Brandolini's law, which means
Brandolini's law. Yeah,
yeah.
I'm aware. Yep.
But we had a break where I was able to. We had like a 3 week break between the next one, and I said these are all the things wrong that he said. I can now regroup because now I know his playbook, and I know he has a weak hand, but he's playing it very well.
Hmm.
So finally, I said, I know I'm going to drop the bomb on him. And I was feeling him out. And finally, I said, well, can you name one geneticist of any repute that thinks the human genome is improving? He gave this blank stare like deer caught in headlights. This long pause. He said, no. And then he changed the subject and he started talking about Dr. Sanford. I said, dude, I could see why you're thinking what to say. It's like, oh, Sal's got me. I know what I'll say. I'll just say, no, real quick, and then start talking about Dr. Sanford and just change the subject, and I'll be saved by the bell, which is what happened.
Hmm.
And this 5 years later, I posed the same question. I said, can any of you evolutionists, just on a Reddit forum, do better than Dr. Dan? When I asked him, do you think the human genome is improving? Can you name one geneticist? They couldn't. What does that suggest? And all this time, Dr. Dr. Dan is ragging on how stupid he thinks genetic entropy is, and he can't name one geneticist that thinks the human genome is improving. So enter recently Kondrashov, and he said the only way we can solve the problem of all the bad in the human genome across the world is to re-engineer it. I'm like, okay, let's see, engineering, that's intelligent design, right? The only way you could solve this problem is through intelligent design. Does it not occur to you? How did the first genome come about? Because natural evolution is wrecking it. You could see it clearly. And the only answer is intelligent design to fix it. How about the only answer to make the genome and the human genome in the first place is intelligent design? And I would put that as a top tier argument. Maybe you can help me figure a better way to say that. We can explore some of my analogies, but that's an example. I won't get any pushback from the mainstream without some of these people looking stupid. Because if an evolutionary biologist presents me and say, oh, genetic entropy is a stupid idea, I'm going to say, well, look at one of your fellow evolutionary biologists who's more senior than you are. This is what he has to say. What do you think? Do you think the human genome's improving? You get them all the time. Just go ahead. If you're on the forums debating with people, I'd be curious to see what the reaction is.
Well, a lot of times people just make stuff up if you try to catch them on that. Like, they just outright lie, and it's like, I mean, I can call you on your lie, but there's nothing I can do to convince you if you're just going to lie about it.
But it's good. What I find it's good batting practice
Mm-hmm.
to see how well, and then you can go back and check your sources. And I'm going to provide the sources. Kondrashov, I have the exact quotes, and people that are. There is a passage, I think, in Romans. It refers to people that are haters of God. I found that really interesting. Haters of God. So I said, these are people, in the modern day, these could be the anti-theists. They deny God's existence, but they still hate the idea. Even though they say he doesn't exist, they hate him. And I see a lot of that in the evolutionary propaganda community. And so they will do, as you say, just lie. But the reason I'm trying to give these ideas is so that creationists can defend themselves. Hopefully they will see through the lie. And I think it should be pretty easy to see that it's a lie or an evasion. Because when I was going through this, my faith was really challenged. I said, I feel like I'm being crushed by Goliath. And, there was always that ray of hope that maybe the creationists were right. And it was because of all the community we had to support us through this. And I feel like I want to give back to that community. So we'll test this out, and then what you can do is you can tell me which arguments you like the most, which you would like to use with your friends. Do you have any prayer groups or outreach groups that you share with? Um, so,
Not at the moment. I've been in, many in and out throughout my life. Kick my desk again. And, but I do have plenty of those communities that I'm still in touch with that I can, come to them and be like, hey, what do you think about this? How's this going to do? So I can always go back to them and get right back in them.
yeah, people more. It's hard to find people that are interested in this stuff. Um, and then I'm also having another project, finding the hidden God, that goes into archaeology stuff that's not so scientifically technical because it is hard.
Mm-hmm.
And I'd be delighted to run some of that by you too. And but I think before I do that, I'm going to continue to prioritize what your questions are and see if I can answer them. And this has been a great interaction. Do you have any? Do you want me to have the last word, or you?
Uh, the only thing that is on my mind to say is I was going to mention something about how you said you had a crisis of faith when having to deal with a lot of those issues. You felt like you're going up against Goliath. And that's just analogous because I sort of had a similar one when I found your channel. And you were a little bit of a godsend because I'm just like, before that, I was already a creationist. I had already seen the merit and the truth in a lot of the arguments. But, there is also the thing of just like, okay, well, they might be right about X, Y, and Z, but maybe it's this or the other one. And it kind of comes from a place, during the time, it definitely came from the place of just like, I need to make sure that I'm not just tricking myself into this. Does that make sense?
Yes.
Like, am I an honest human being? Like, do I actually believe this, or am I just tricking myself into this to make sure what I believe? And I don't necessarily think any of the evidence on the evolutionary side was so persuasive. It was more along the lines of just like, you make that honest assessment like you did, where it's just like, okay, God is not as apparent as we would like him to be, and why not? And that's where all the real, any of the actual weight came down into it. And then I found you, and you pretty much voiced my thoughts 100%, and I'm just like, thank you, Lord. And, it didn't immediately solve it, obviously, but your channel was very thorough and very honest in terms of getting to the nitty-gritty and the actual truth of any of the technical process that is the core of the evolution creation debate, is what is actually happening. We're cutting through all the equivocation, cutting through all the PR. What's the reality of the situation? And you're able to not just make the claim, but thoroughly show that, hey, the reality of this topic, X, Y, or Z, supports creation. I mean, sometimes it's ambiguous. Sometimes the evidence doesn't go one way or the other, and it could, sometimes it's trying to make an argument. The sky is blue, therefore evolution is real, or the sky is blue, therefore creation is real. It's like that does not work. That's not how that is. That's a non-adjudicating matter of inquiry. I'm simplifying, obviously, but that's how it would go with a lot of them. But I just wanted to make that parallel because when you said that about your experience, I'm like, wow, that feels about right. And that's all I really wanted to say on it. So now I think you should have the final word, as the teacher and mentor of this learning session here.
Well, thank you. Glory and honor to our Lord and Savior Jesus Christ. There's been a succession of, people in the faith. Our forefathers have passed on the faith to us. I'm very grateful for the creationists who fought the battles when the battle was really looking really, really bad. I mean, we had an example, in our 2 hours, we've experienced a battle that had been ongoing for 40 years on the question of Nylonase. And it seemed like we were fighting Goliath. And so this is comforting for me to reflect back of how dark everything looked at one time and hopeless. And this is also around the time I nearly lost my faith. I had bouts of agnosticism and just stopped believing. And it didn't last long. And it's a joy to be able to share my journey in my recovery when I began to see. Um, it says in Proverbs 25:2, it is the glory of God to conceal a matter. It is the glory of kings to search out a matter. God does not make it easy to find the evidence of himself. That is to his glory. It is to our glory to search out what he has concealed. He has concealed it, but in a way that is not so hard to uncover. If he really wanted to be so hidden you wouldn't discover him, he could do that. He's chosen to make it hidden just enough so that the people who really want to find him will find him. But the people that want to close their eyes to him, he'll make it really easy for them to turn away and walk. And now I'm seeing that. And so I'm grateful for people like Anthony who are seeking after God to want to know his works and him better. And it says in the Old Testament, I, I will declare the works of God. So that is what we have done today. Glory and honor to our Lord and Savior. And until Lord willing, we meet again. Take care and God bless you all.